Hey class, let's continue our discussion on viruses with specific discussion on how we cultivate viruses. There are two primary techniques for cultivating viruses. That's the in vivo and the in vitro technique. Both of these techniques depend on living cells. Viruses cannot reproduce without a host cell. The in vivo technique, or in life, think of Viva Las Vegas, viva meaning life. In vivo, or in a living organism method, is when we have it in an laboratory-bred animal or an embryonic bird tissue. Chicken eggs are a very popular technique for the in vivo technique method. Or we have the in vitro technique, where we have cell cultures being reproduced and then infected with the virion particles. The in vitro or in glass technique is typically going to be the in test tube or the in the petri dish technique. And there's a third one that is much less popular, typically because it's not as reliable from a results perspective, and that's the in silico technique, which your textbook doesn't spend a lot of time talking about. That's the computer simulation technique, or silicon computer chip technique. Usually the in silico technique is going to be done before the in vitro and then before the in vivo technique. The in vivo technique is the gold standard. In vitro is pretty good, not quite as reliable as in vivo, and then in silico is that red-headed stepchild that can be done. It's a nice supplement to your article if you're publishing or to your study, but generally speaking, the data is not very reliable in and of itself. When we go through viral cultivation, because we need to isolate and identify viruses from a clinical specimen to make a diagnosis, we can use these viruses that have been isolated for vaccine preparation, or we can do more detailed research on the virus life cycle and effects on the host cells. Once we can isolate and cultivate the virus, we can better understand the virus and combat that virus. If we use a live animal or an in vivo technique, we are going to use a specially bred strain of lab animals. So we'll have some white mice, some lab rats, hamsters, guinea pigs, rabbits are going to be the usual choice for our live animal cultivation. Every once in a while we'll use an invertebrate, such as an insect or a non-human primate. The non-human primates are rarely used, generally speaking, because they are so expensive to maintain in a laboratory setting, and there's a lot of regulatory and ethics paperwork that needs to be filled out with the use of primate studies. Insect studies or invertebrate studies are rarely used because there's not much interest in them. There's not much government funding associated with invertebrate studies, because invertebrate systems differ enough from vertebrate systems that many viruses that will affect invertebrates will not have an effect on vertebrates. So if that virus affects a beetle, it's probably not going to affect a human. And there are a few that cross over, but those are few and far between. These viruses exhibit very notable host specificity to certain animals and can propagate viruses, and because of that host specificity, certain animals are capable of better propagating the virus than other animals. In other words, some animal species are more susceptible to viral infection and can produce more virion particles than other animals, which are naturally more resistant to viral infection. Bird embryos are commonly used because they're still emulate that animal system, that in vivo system, and they are completely self-contained. They have all the food, they have all the water right there. They're very, very easy to work with. They are the easiest animal system to work with in a laboratory setting. Also, generally speaking, there's fewer ethical boundaries associated with working with a bird embryo because the bird hasn't been fully born yet. When you work with fully born, fully developed invertebrate animals like bunnies or guinea pigs, or especially with primates, there's going to be a lot more paperwork associated with that. Bird eggs are also excellent because they are naturally sterile in their internal environment, and the yolk contains all the nourishment. Also, there's the albumins in the egg white as well. When we use bird embryos, chicken are the most common, but we can also use turkey or duck eggs. That virus will be just injected directly through the eggshell. You'll take a small power drill, make an itty-bitty hole in the shell, or you'll crack off a small section of the eggshell and then inject the virus through the membrane of the egg. If we use the tissue culture technique, you're typically going to reproduce that animal cell in a test tube or in a tissue culture dish, many times these tissue cultures are going to be cancerous or precancerous strains of tissue culture that are capable of reproducing indefinitely. Particularly when you're working with human tissue culture samples, they are notoriously finicky for dying. You need to maintain the correct extracellular environment for those tissue cultures to grow, and unless you're constantly filtering new media over the extracellular environment, those tissue cultures will die if you don't check on them, typically every 12 hours. Cell cultures need to be grown in a sterile chamber with that special media. Most of these cells are going to form a monolayer or a single layer of cells that can be supported for viral replication. If the wrong growth factors are introduced into the growth medium, you'll have stratification of the cells, and then the cells at the basement membrane or basement layer that are away from the growth media will begin to die off and have necrosis and decomposition set in. And then everything dies in your culture. Tissue cultures are a pain. If we look at the degradation of the cells by the virus, this is a way that we can monitor the growth of the virus. So if we're working with a tissue culture technique, we can monitor how virulent the virus is by measuring how many of the cells, visibly that we can see, have been killed off by the virus. Especially if we're working with a Petri plate and bacteriophage, and that bacteriophage is introduced to a Petri plate covered in bacteria, that bacteriophage can spread out and kill the bacteria in a growing circle. So there'll be this growing circle of dead bacteria on the Petri plate that will manifest itself typically as a clear area on the Petri plate or a well-defined patch on the cell sheet if we're working with a eukaryotic specimen. If we analyze the plaques underneath a microscope, you'll see the cytopathic effects or the pathogenic cell effects of these viruses on the cell cultures. Here we can see healthy cells on the left. Each of these cells are separated. We have one nucleus per cell, and then here we can see some cytopathic effects over here. We can see cells that have been grouped together, so we have multiple nuclei in one cell, and then we have a lot of clear space as well, so we can see that this has very clearly been infected by a virus. This is abnormal histology, this is pathogenic histology, and this is healthy histology on the left. The same technique that we use to grow viruses in culture can be used for animal cells, can also be used for bacteriophages. I alluded to this just a little bit ago, where we take a Petri plate covered in a bacterial species, have a lawn or a carpet of bacteria growing, and then drip, drop, drip, put little specimens of our bacteriophage on the Petri plate, and then we'll have that growing clear ring of no bacterial growth radiating out from where we introduce the bacteriophage. As a new cell gets infected and dies from the bacteriophage, it releases more bacteriophages, which infect adjacent cells, which then kill those adjacent cells and affect the next layer of adjacent cells. And as the plaque gets bigger and bigger, bigger plaques mean more virulent pathogens, smaller plaques mean less virulent pathogens. So, concept checked. Which of the following is not an in vivo, an in-animal method for culturing animal viruses? An embryonic chicken egg, or embryonated chicken egg, man, that's a wordy term, a guinea pig, a dog kidney cell culture, a white mouse, or all of these choices are in vivo methods. Remember, in vivo means in the animal, not in as opposed to in vitro, which means in glass. So pause the video and rewind it or flip back in your PowerPoint to look up the answer. 1, 2, 3, 4, 5. The correct answer is D, dog kidney cell cultures. All of the A, B, and D represent a whole organism, whereas C represents a specific cell type from a specific organism. So A, B, and D are in vivo techniques. C is an in vitro technique. If you have any other questions, please post them on the discussion board or shoot me an email. Happy studies, class. Bye-bye.